Hello, I am John Martin. The message of this video is that stealth-adapted viruses transmit renegade genetic sequences. Let me put this into context. We generally understand viruses are genetically distinct from the genes in cells, bacteria, and fungi, even though in antiquity there have been some limited genetic exchanges between cells and viruses. One example, the retroviral sequences that are present in the human genome. What the research has shown, it's identified a new category of viruses that, one, evade the cellular immune system, and two, transmit a range of unstable cellular and bacterial genetic sequences. They can transmit them between individuals and even across species. The idea of sequences hitching a ride with these viruses leads to the term renegade. So I refer to these viruses as stealth-adapted viruses with renegade genetic sequences. They're derivatives from regular viruses. We know they cause many chronic illnesses, not only chronic fatigue syndrome, autism, but several neurological diseases. Primarily, the brain is uniquely susceptible to illnesses caused by limited localized damage from these viruses. But what we're also facing, the fact that the transmitted cellular and bacterial sequences could be inducing a whole new wide range of chronic illnesses that would not initially be thought to be infectious, but in reality will be caused by the transmitted renegade sequences. There's a real urgent need to culture and further characterize these viruses, and then to develop the therapeutic approaches, not on enhancing the immune system, but rather utilizing what I call the alternative cellular energy pathway. Now, again, it may be helpful in understanding this research if I go back to the beginning when the research began. I had joined the University of Southern California as a tenured professor of pathology in late 1985, and in 1986 I became aware of three events. One was description of a mystery illness by Drs. Dan Peterson and Paul Cheney, who were working in Incline Village in Nevada. It was really the same illness had been described before under the term myalgic encephalomyelitis, but when it became the focus of attention of the CDC, the name chronic fatigue syndrome was applied. The second development in 1986, Zaki Salahuddin, working in Bob Gallo's laboratory, discovered a new herpes virus that came to be known as human herpes virus 6. The third development was that of a highly sensitive molecular diagnostic assay known as polymerase chain reaction. We know of this assay because it was the mainstay of the early diagnosis of COVID-19 infections. So back then in 1986, I said as a research project, I would learn the PCR assay so that I could test chronic fatigue syndrome patients for human herpes virus 6. I was fortunate to learn about Dr. Jay Goldstein, who had been treating myalgic encephalomyelitis patients in Anaheim, California. He taught me a lot about the illness and kindly provided samples for me to develop the polymerase chain reaction. Let me now give some little bit more details about the PCR assay. The two lines on above, double-stranded DNA. There are two strands of DNA, one going in one direction, the other going in the opposite direction. These strands are held together by hydrogen bonding but they can be separated using heat. The idea of the PCR assay was that one could look along a long stretch of DNA and identify a small target sequence, 200 or 300 nucleotides long. If you knew the sequence in that area, you could design probes so that when you separated the DNA strands, you could add these small probes into the reaction, and they would bind the targeted DNA sequence, one at one end, and that would then be extended towards the other end, and conversely, it would be flanked by a second primer nucleotide sequence. So that reaction could occur using an enzyme called Taq polymerase, which would then extend the two sequences so that essentially from the first cycle, one could take that small stretch of DNA and amplify it so that you would have two copies. What was nice is one could then repeat the next cycle, reheat the formed DNA, add more primers, or have the primers that were already present, would bind again, add the enzyme. You would then get, from the two copies, you get 4 copies, and then 8, 16. So one can see how one could, over a 20 cycle, 30 cycle period, provide a very large amplification of that particular DNA sequence that was being targeted. When you made the DNA, you could then analyze it. One approach of analyzing it was based on size. Agarose, is a product from seaweed, allows DNA to move through it slowly. It will move through it depending upon the size of the DNA. If the DNA piece is relatively small, it will go through fast, and in the gel will be down towards the bottom. If it is a larger piece of DNA that was amplified, it would stay up higher. You could identify the DNA in the gel because it would stain with various dyes, but a common one, ethidium bromide, as a stain because it's fluorescent and easy to see. You could also, as shown on the left side of this, use known size DNA as markers. So this would give you an indication. What I can point out in this slide is that we're looking not at a single PCR product that one would anticipate if you use the exact sequence, but rather we're seeing examples in different assays of multiple products being formed. This is because what I learned, since we did not find specifically human herpes virus 6 as a reliable marker or consistent marker in chronic fatigue patients, what we realized is that we could adjust the PCR assay so that the regular use of high stringency PCR that had specificity to it, we could overcome that by making primers and allowing the primers to bind using lower temperatures and different conditions, almost like a fishing expedition, to see if some of the sequences might bind, might amplify the DNA. We set up those assays, and then whatever DNA was formed, we could further characterize it using a hybridization reaction. And what this is again producing a small synthetic section of DNA that would then take the PCR products, heat them to separate the strands, and then react with these small pieces. And if there was some binding, then it would show up. A useful marker is to make the probe radioactive. So using just X-ray film, you could get a black area where the radiolabeled probe was bound. And generally, we could take all of the different PCR products that have been separated and let them bind to a nitrocellulose membrane. So they would be fixed, add the probe, and then see if there was any radioactivity. This shows 56 assays that we performed, and you can clearly see the wells. These are material added in different wells to an ultracellular sheet, and then the sheet was exposed to the detecting probe. But you can clearly see some of the assays are positive and many others are just no signal. So it became quite easy to distinguish a positive assay from a negative assay. The other technique we utilized is to clone PCR products. And for this, there are plasmids that infect bacteria. And they have a number of genes, some of which are not essential, and you can remove those genes and then substitute DNA that you want to amplify in the plasmid. So there's an example on the top there of a plasmid, the genes that are non-essential taken out, that would grow in bacteria, and those bacteria would stay susceptible to an antibiotic. On the lower one is when we insert a piece of foreign DNA, could be cellular DNA or PCR product, into that plasmid, and then put that plasmid into bacteria, we can select those bacteria because they become antibiotic resistant, and the plasmids from those bacteria will contain the insert. And then we can sequence the DNA in the insert and gain an understanding of the DNA amplified in the PCR assay. So here's the summary of the early studies from 1986 to 1990. We had positive PCR assays in several interesting, complex neurological patients. And in retrospect, I probably should have focused on those rather than getting into the more politically complicated chronic fatigue syndrome. But certainly, up to a third of chronic fatigue syndrome patients were giving us clearly positive PCR assays, providing I dropped the stringency way down. I used probes that would amplify all types of herpes viruses, whether it was herpes simplex, Epstein-Barr, human herpes virus 6, broadly reactive primers. And it was clear that a good number of chronic fatigue patients had evidence. A real insight came when we had an opportunity to look at brain biopsies, and there was one particular patient, a schoolteacher from Palm Springs, who provided the opportunity to look at tissue for a positive PCR assay. And this was interesting because, as I'll show, the biopsy of the brain did not show any inflammation, which is the normal hallmark of an infection, and yet it was PCR positive. But let me just go back to one of the examples of a neurological disease. This was a child who was born at a local hospital here in Los Angeles. The child had what might ordinarily be considered like a cytomegaloviral infection, but in the sense that the liver was enlarged, the spleen was enlarged, the choroid plexus in the brain was damaged and there was bleeding. The child was not thriving and then developed a hemorrhage within the brain. They had to put a shunt into the brain, and that provided the fluid from the brain, cerebrospinal fluid, that would be tested. So I worked with the clinician there, and what this shows, the positive controls, the top A and B, then this was cerebrospinal fluid samples, C, D, E, and F, and then the negative controls, other parts of the gel. Very clear positive that here we had a child where there was clearly an infection, and yet again, it didn't have any inflammation. So that was, as it were, that child. This is an example on a brain biopsy where the band shown in the high up in the gel is a positive PCR on the brain reaction, on the brain tissue. Now, the patient whom we took special interest in was a schoolteacher. Her illness began as a chronic fatigue syndrome. She lost her job and she obtained a secondary job as an elementary teacher, but she couldn't cope. She couldn't remember the course. This was her efforts at drawing a clock and a house. And it was of interest to be speaking with her in the sense that she could identify a tie, but a shirt, but certainly not a button. She was at a loss of words. Very sweet lady, and her real concern, she was no longer able to communicate with her husband. Now, in further analysis of this by the neurologist at the University of Southern California, the MRI showed enhanced white areas around the ventricle parts of the brain. It was unknown what illness she had. It could have been a cancer, a brain tumor, could have been some infection. So the neurosurgeons offered to do a needle biopsy of a very small amount of that tissue, and then I had the opportunity to examine that tissue. This next slide shows the tissue actually removed by the biopsy, submitted to the laboratory. What's striking is that it's essentially pink in appearance. There's no inflammation. If there was inflammation, it would essentially be more bluish because of the lymphocytes and polymorphs and macrophages that would be infiltrated. On higher magnification, you can see cells that have abnormalities, and I'll provide more details about each of these slides in a later seminar. But it's interesting. There's no inflammation. There are cells yet show a degree of damage. There are some cells that have syncytia, so that several nuclei have fused together within the one cell. Cells have fused, so there's multiple nuclei in the cells. We did electron microscopy on the tissue, and it too showed abnormal cells. What this shows are these vacuoles, open areas. On the top of the slide, you can see some residual material, which is lipid-like material, like a foamy cell. There are also these unusual appearing structures, which we now know to be these alternative cellular energy pigments, not shown on this slide, but when we could identify mitochondria, they were very deranged in this. So we clearly had an example of cellular damage, positive PCR assay. We knew there was some virus involved, but it was not causing inflammation. And that led to the concept of like a stealth virus. If we take the analogy of the stealth bomber evading the radar detection, what we had were viruses that were evading the recognition by the cellular immune system. So with this in mind, we went back and made a very intense effort to culture viruses from PCR positive chronic fatigue syndrome patients. I had been trying to culture for some time, and in fact our laboratory was routinely culturing cytomegalovirus from renal transplant patients from a major hospital in Los Angeles. But we put effort in, and I think the first culture took 6 weeks or so to turn out positive. This was the PCR positivity in the patient. The 3 top dots are the positive control, see the patient, and then the negative controls. What this shows is a culture we don't normally stain, we just look under the microscope, but we can take the cells from the culture, stain them so that we can see. And what this shows are relatively normal cells still, small spindle-shaped cells, but you can see these foci developing in the culture that were clearly a positive viral culture occurring in this sample. With this patient whom we tested, the cultures could go even further and develop into these cells that had multiple nuclei, foamy, vacuolated. So we had a very clear evidence of a cytopathic effect developing in the virus from this chronic fatigue patient, and that became the opportunity to do molecular studies. Just in summary, and this will then lead to the publications, what we had then was the opportunity to isolate the low-stringency PCR-generated product or products. We'd be able to insert the products into plasmids. We could select the plasmids with the inserted PCR products, and then we could do DNA sequencing on the inserts. Once we had that, we could then go to what's called GenBank, which was a repository of all publicly available sequences, and see how our sequences would match. What we also could do is take the PCR product and radiolabel it using P32 as a strong radioactive marker, and then with that, we could go seek out the whole virus in the cultures. If we had the virus, or when we did have the virus, we could cut it into smaller sizes that could be inserted into plasmids using what are called restriction enzymes. Then we could sequence the plasmids that were detected using the radio-labeled PCR product to confirm that it was present in the virus. We can also clone the entire viral genome so that we could get additional sequences. Finally, now that we had sequenced, we could go back to repeated samples of blood from the patient and reconfirm that the virus was persistently infecting the patient. All of these data were put together in a major publication called Cytomegalovirus related sequences in an atypical cytopathic virus repeatedly isolated from a patient with the chronic fatigue syndrome. What I'd like to do is set up a second video beginning with this paper to go through and show you the results we obtained using the molecular procedures that I just described. Thank you very much.